All right, hello and welcome to this webinar. I make it 3 o'clock, so I'll start with a little bit of general information. So this is our first webinar from Fluidic Sciences. It's going to introduce the technology microfluidic diffusional sizing and look at some of the ways in which our machine learning guided assays can help accelerate affinity measurement in our in-solution assays. We expect the webinar will be about a half hour long. If you have any questions at any point during the webinar, please just drop them in the chat. There should be a question and answer type box there for you. And at the end of the presentation, my colleague Nadia will work through those questions. If we don't have any time to do the answers live, then any of the questions that we don't get around to, we'll come back to you directly to provide an answer. So I think with that, we're about ready to get started. So my name is Sean Devenish. I'm Chief Scientist at Fluidic Sciences. I've been working with the technology microfluidic diffusional sizing for quite a while now. And it's a pleasure to sort of take this opportunity to introduce the technology and its applications to you. So sort of, I guess it's nice to start at the beginning, and where we started when we first were thinking about developing this technology is really where are the challenges in protein interaction analysis. And for anybody who's worked with proteins, many of the kind of features on the slide, many of the content in the slide will be very familiar. So, proteins are very complex molecules to work with. Some of them are easy and many of them are very difficult. They are highly diverse and frequently heterogeneous. And so while existing technologies have served us pretty well to date, there is still a number of proteins and proteinaceous species that are very difficult to analyze for a wide variety of reasons. And so our motivation behind developing the technology of microfluidic diffusional sizing was really to help to address some of these more difficult to work with samples. So this could be because of the target itself, the protein, or also because of the matrix in which that target is presented, can also create a lot of problems. So when we were thinking about, where do some of these challenges arise from, and we were really focused on how existing technologies, or the vast majority of existing technologies work, is really based on detecting interactions at a surface. Now, these have done a great job and have really moved forwards, biological research a very long way over the past years. However, there are consequences of working at a surface. And so what we see, for example, when working at a surface is you can have binding of background molecules directly to that surface. And obviously, if you're just detecting interactions at a surface, this would be detected as a binding interaction. Likewise, your analyte can bind directly to the surface without being mediated via the target. So again, this is an artifactual interaction here. The target itself can potentially be modified by binding to the surface. So we can either have conformational changes induced by being affixed to the surface, or potentially steric impacts of being close to this sort of very large and highly blocking interface. And finally, you can also get, for the case where you have multivalent analytes, you can get avid binding as opposed to affinity binding. So these sorts of Velcro type interactions where the apparent affinity is greater than the true affinity. And of course, against all of these sort of artifactual interactions, we have the interaction we're actually interested in. And so if we can simplify the system and get rid of all these artifacts, then that will open up our analysis sort of scope quite a long way. And so that's what we've decided to do, is we've developed a technology that works solely in solution. And of course, by removing the surface, all of these artifactual interactions disappear. And so then we can really spend our time focusing on the interaction that we're actually interested in. And so this makes it much easier to work with things like multivalent, polyvalent targets, to work with molecules that are sensitive to their environment, such as intrinsically disordered proteins, and to work with things like highly complex matrices. And I'll present some data on these sorts of interactions in the coming slides. So before getting into that, though, I think it's best to step back a little bit and say, okay, so we developed this technology called microfluidic diffusional sizing. What does it actually do? What does the data look like? So what you're looking at here is a case of a simple binding interaction affinity curve. So we have, first thing to note is that our vertical axis here is hydrodynamic radius. So the technology itself actually measures size. It measures the size of a fluorescently labeled particle. And then as we add a binding partner, the size of that fluorescently labeled particle increases in solution as it starts to form a complex. So we can see here we have our fluorescent labeled particle at this end of the curve is essentially on its own or has no binding partner. And as we titrate in our binding partner, this binding interaction increases until it saturates at the high concentrations of binding partner. Now, by fitting this curve, we can get the KD. This is obviously the key parameter we're primarily interested in. But we also get, alongside that, these quality control measures shown here. So the RH free is the size of our free antigen. And this tells us what's the quality or reports the quality of our antigen. So perhaps we can detect whether that antigen has changed in size since we last measured it, since we produced it perhaps a few months ago and it's been stored in the freezer. Maybe it's aggregated and gotten larger or been degraded and gotten smaller. We can also ask the question whether this size is consistent with what we expect for the antigen we're working with. So we have some nice tools on our website to predict the hydrodynamic radius of a molecule based on either the molecular weight or the structure. And so we can compare that against what we measure and see if there's any significant disparity there. Likewise, RH complex is the size of the saturation complex that we're forming. And again, we can ask the question, is this consistent with what we believe we know about what's happening in this reaction? And again, if this diverges significantly from our expectation, then we need to go back and have a little look at what's happening there. So in a single run, we're picking out the KD, this critical parameter, telling us about the binding affinity, as well as these QC measures. It's very easy to do. So now I want to just extend beyond that, and this is getting more towards this idea of these advanced assays using machine learning guided tools, which I'll talk about the tools themselves subsequently. But first, I just want to show what we can do with these assays. So the first is our sero affinity and concentration assay. And so this is really focused at looking at serum or at antibodies in serum. Now, of course, we can substitute serum with any other complex matrix. We would probably argue that serum is about the most complex matrix there is. But this could work equally well with other biofluids, or, for example, cell culture medium, if that's a matrix of interest. Now, the motivation behind this is to really move beyond ELISA. So you can see we've got here some matched samples measured by ELISA on the left and using our SAFCON assay on the right-hand side. So ELISA obviously is very widespread and very heavily used, and in some sense is great technology. However, it does give you a somewhat semi-quantitative, perhaps qualitative type ranking without really capturing what this specifically means. So if somebody else repeats the ELISA in a different lab, they may get a slightly different ranking order. They'll almost certainly get different absolute values. So it doesn't really, it's not a quantitative measure of anything, and it captures some amount of affinity, some amount of concentration, largely concentration, and to a certain extent a bit of the off-rate kinetics as well. Using our SAFCON assay, we can take these same samples and actually start to understand them according to their fundamental parameters of affinity and concentration. And so you can see here, for example, if we look at the top rank sample by ELISA, what we see in our SAFCON assay is this one does indeed have a very high concentration. It's by far the highest concentration in the cohort of samples. However, it has quite a low affinity. And so if it's affinity that you're interested in, then choosing sample 1 as your starting point would be a losing proposition. Whereas samples 3, 4, and 6, so sort of in the middle of the range here, actually represent those with the greatest affinity. So, this highlights immediately the benefit of moving from a sort of a single measure that captures a bunch of different parameters in a semi-quantitative fashion to an analysis that captures fundamental properties of the sample in a single assay. So it's very sample efficient as well. So just draw your attention down the bottom here. This only needs 60 microliters to carry out this assay. So really you can make a lot of your sample, and that's one of the benefits of working with microfluidics as we do. The second example here is really looking at challenging targets. So disordered proteins, intrinsically disordered proteins, and macromolecular species. So the protein of interest here is called alpha-synuclein. It's the causative agent of Parkinson's disease. And as a monomeric protein, it's intrinsically disordered, whereas in the course of the disease, it forms these higher order structures or aggregates that in the first instance are described as oligomers. These are clusters of several alpha-synuclein molecules. And then these continue to grow on to form fibrils, so the hallmark of the disease of Parkinson's disease. So all of these are difficult to work with on using surface technology. Intrinsically disordered proteins tend to adopt a fold when immobilized on a surface, and these larger macromolecular species can be difficult to immobilize, and even when immobilized, there's a real risk of them having orientational effects. So are they lying down or standing up? And then obviously steric considerations when you have a whole surface of the molecule blocked by the sensor surface. And so what we've done here is we've measured the affinity of an antibody called 4D6 for these 3 separate species. And so what we see is as we look across the transition from monomers to oligomers to fibrils, there's no appreciable change in the affinity of that interaction. So it's consistently around 10 nanomolar. So this tells us the epitope is not really changing in its structure as we're forming these species. But you can also see here there's a second line, and this is where we're capturing the stoichiometry. So in this case, we're directly measuring the number of alpha-synucleins interacting with the antibody. So for the monomer, we can see we have 2 alpha-synucleins interacting per antibody. For a small protein interacting with an antibody, this is what we expect, with 1 binding to each arm. Once we get to the oligomers and fibrils, this ratio increases, and the reason for this is because we have essentially a number of alpha-synuclein molecules that are traveling with one that presents an epitope. So in other words, for the oligomer, we need to have 5 to 6 alpha-synucleins present in a cluster to present a single binding site. And in the fibrils, this ratio increases to 7 to 1. So we can see that as we are increasing the size, we're obviously having a number of alpha-synucleins where the binding epitope is buried inside this macromolecular constructs. But it's also worth noting that these actually still represent quite high binding ratios compared to those seen for some other oligomeric type species. And then finally, in this section, I'm going to talk about membrane proteins. So obviously, membrane proteins are a very important class of therapeutic target, well overrepresented in the existing pharmaceutical world. Now, the challenge of working with membrane proteins is that obviously solubilizing them, they need to have this membrane detergent or membrane or detergent environment. And even just determining what detergent is suitable for any particular membrane protein is really a whole project in itself. So solubilizing them is difficult, purifying them is also difficult. And so what we've done with this assay is actually bypass both of those steps. So the way the assay works is we take a cell culture and we harvest the membranes from that cell culture. The membrane is then directly solubilized using polymers that are able to break the membrane up into small pieces and then stabilize those small pieces in structures called nanodiscs, also known as SMALPs. And then this crude library that derived directly from the intact membrane is the input to the assay. So there's no purification and there's no need for detergents or detergent screening or anything like this. And so we can then assay directly for our membrane protein of interest in this membrane protein library, and we can determine the affinity of an interaction against that membrane protein, and then we can also determine the copy number of that membrane protein. So you can see here we're determining the number of our target present per cell in the starting culture, which the whole experiment started. So again, very powerful and really nice to be able to get to actual copy numbers of membrane proteins. So very powerful. So these are some really nice assays, and now I just want to talk quickly about how we do it. So essentially, these are the products that we have on offer. So there's two parts to this. There's Fluidity Insight, which is our analytical platform and really is the engine that enables these assays, and Fluidity 1M, which is the instrument itself that provides the measurements. So I'll start with Fluidity Insight. Now, as you might imagine for these assays, they're somewhat complex, and so, if we think about when we're trying to determine the affinity and the concentration, if we walk up to a sample when we don't really know anything about it, say, for example, a serum sample, we don't know anything about it, but we want to know the affinity and we need to know the concentration as well. So if we take a binding curve and fit this 1,000 times for affinity and concentration, and this is the result. So what we see is that the two properties are correlated against each other. So we can essentially trade off affinity against concentration. We don't really know what either of these properties are. So any of these results are equally good fits to this data. So this creates a problem. However, because we're working in solution, we can solve this problem by running a second binding curve at a different concentration of our probe molecule, as shown here in this pink curve. And then what we do is we fit these data together in a single global fit, and then we're asking what combination of KD, or of affinity, and concentration uniquely fits both of these curves, or all of these data. And you can see here, when we fit these two curves together 1,000 times, we get good convergence. So all these results land on a quite focused region. And so that's really the core of the assay. And what happens on Fluidity Insight. So Fluidity Insight takes care of all of this for you. Now, as you might appreciate, that one of the big challenges here is that it's actually quite hard to know intuitively what extra kind of data points you need to add into the experiment to get the result you need. Now, that's one of the really nice things about the platform, which we've managed to put together, is it has an experimental guide element. So this sort of shows the workflow for one of these experiments. What you do when you walk up to the experiment is you take your sample, and it doesn't really matter what the sample is, you run a standard set of conditions. And then these first set of results are fed into Fluidity Insight, which carries out the analysis and does the recommendation on where to proceed. So you can see that kind of blown up at the bottom here. So this would be an example of an initial analysis. So you can see we still have quite a poorly constrained result with a long tail off to the right-hand side here. Now, obviously, as an experimenter, looking at this, it's fairly opaque, a little bit hard to know, well, what actually do I need to do to improve this situation? And so what the platform also does is organize the potential experiments and show you where to go hunting next. So this is an example of the output. We can see what we've already done, and then the algorithm is recommending that we work with something in this bottom left corner in this case. So a low concentration of our target and a low concentration of the probe. So then we jump back out from here and we come to our second experiment. And so we would run one or two data points from that recommended region, and then feed those additional data points back into Fluidity Insight, where those results are combined with the previous data set, and the fitting is run again, and then you can see the final results. So you can see we've very quickly got a very nice constraint on that, and we've got a well constrained result. And so it's really very straightforward. So in essence, I mean, the beauty of this is you actually don't need to know anything about biophysics. You just need to be able to follow some fairly straightforward instructions to be able to get these powerful results. And then just to quickly touch on the instrument itself. So this is the technology that allows these measurements to be taken. Then we have a benchtop reader and then a consumable that contains the microfluidics. So the benchtop reader itself is entirely fluidic free. It's actually quite powerful in that microfluidics is it's a fantastic technology. The key challenge with microfluidics is the microfluidics themselves, trying to maintain an instrument with microfluidics. But what we've managed to do is actually push all the microfluidics in this unit down onto the consumable. So the benchtop reader itself is very robust, contains no microfluidics. It has two optical channels, one in the green and one in the red. So you've got a lot of experimental flexibility there, as well as having temperature control. So even on the coldest of days, such as today, you're going to get consistent results with what you see in the middle of summer. And it's a self-standing machine, so it has a touch screen, no need for a computer, so it's very economical on bench space and sort of very intuitive to use. Looking at the consumable, obviously it has a dry interface to the instrument, so no fluids cross into the instrument, which also means you don't have to worry about cross-contamination of the instrument, which is also another benefit there. There are 24 circuits per plate, so three sets of 8, and you can use part of those. You can use part of the plates. You can use as few or as many circuits in any particular run as you like, because there's an RFID tag in the chip that gives traceability, but also allows you to know which circuits are still available for a measurement. And as you can possibly guess, looking at the layout of the chip, it's multi-channel pipette compatible. So loading the plate only takes a couple of minutes using a multi-channel pipette. So for the last few minutes, I just want to take a deeper look at antibody characterization. So this is looking at a few published papers that we've put out and kind of bringing them together to look at how we can look at antibodies. So this research shown here is some work we did with the lab in Switzerland. This was during the COVID pandemic, and we're looking at survivors or people who post-COVID infection and characterizing their response against the wild type virus. And so you can see here we've got it's about 30 individuals, and we have the vast majority of them were not hospitalized, and 3 of the individuals were hospitalized. Now, the other thing you can see here on this graph is this sort of gray region on the bottom left corner, and this is the region in which we would not expect, because of the biophysical properties of binding, to see binding. So if there's an individual with an antibody response lying in this region, then they won't be mounting an effective binding response against the virus, and also we are not able to characterize that response using our technology. What we can see when we look at the data is, first, there's quite a broad range of affinities. The concentration range is somewhat narrower, but we have about a 4 log range of affinities. The majority of people sit relatively close to this non-binding regime, so they're not mounting particularly strong antibody responses. However, there are a few lucky individuals who've got very tight binding antibodies. Worth pointing out as well, all of these measurements, again, are made without any external calibrant or reference samples. That's one less thing you have to worry about, is the quality of a calibrant. So we were actually able to collect extra samples from these 3 hospitalized patients and look at those in a bit more detail. So because these patients have been hospitalized, samples have been taken over time, and so we had access to these longitudinal samples. The numbers next to the data points here show the days after onset of symptoms. So what we can see is that up until about 10 days, these patients are not mounting an appreciable immune response, which is quite possibly why they ended up in hospital. After that, we see the responses pop up, but what we see over time is evidence of an increase in concentration, but no real increase in affinity. So obviously that's what we'd hope for, is affinity maturation process to take place, and for these patients to develop high affinity antibodies. But what we're seeing, at least in this relatively narrow time window, is essentially a brute force type response of the immune system. Just make more of the antibodies that are there, and perhaps we can worry about affinity another time. So, even after these almost 2 weeks and hospitalization, these patients have not developed particularly high affinity or good quality antibodies. And finally, we were able to then expand the study yet further by collecting samples from a number of different centers. So we ended up collecting 74 individuals, and I guess it sort of tells the same story, but just in a larger scale. So again, we can see a very wide spread of affinities, with the range being sort of, again, kind of 4 logs there, where the concentration is more narrowly focused. And again, we can see this clustering sort of not far from the non-binding regime. We did add to this, however, a pooled serological standard. So these points shown as circles are the World Health Organization serological standard, and measured against 3 different variants, so wild type, Delta, and Omicron, where the squares are all measured against the wild type virus. And what we see as we move from wild type to Delta for this pooled sample is a decrease in affinity, about a 2-fold decrease in affinity, but no change in concentration. So all the antibodies are still binding, they're just on average binding a bit less efficiently. When we move to Omicron, we see a step change in concentration as well. So as well as another approximately 2-fold decrease in affinity, there's also about half the antibodies cease binding altogether. And we see this represented as effectively a decrease in the concentration of the antibodies. And so this sort of tells us the story, and on reflection, helps us understand the transition from wild type to Delta to Omicron that we saw in terms of human infection. So this is my last slide. Just hopefully I've given you, in this brief talk, a bit of an overview of the technology, and have shown you how the microfluidic diffusional sizing is a direct way of measuring binding. So because we're looking at the size, it's a very direct and straightforward, easy to understand technology and approach for characterizing interactions. It's a very simple technology, so there's really nothing more to it than mixing and measuring. It's highly versatile, so it can work with both high and low affinity samples, and can work as equally well with simple or complex systems, as evidenced by, for example, the many serum samples that we've analyzed. It is very economical with samples. So, we know that samples are always precious, even if it's a purified protein. These things cost money and time to get hold of. And so we're able to maximize the amount you can get out of your samples. Very powerful due to these multi-attribute results, as seen in those last few slides there. Really looking at size, binding affinity, stoichiometry, and concentration. And finally, it's unbiased, so there's no need for calibration. And as I mentioned previously, you don't really need to know anything about biophysical or biophysics in order to be able to use this biophysical technology. So with that, I will ask Nadia to let me know if there's been any questions.
Thank you, Sean. Yeah, there is a few questions here that we have got a couple of minutes to answer. So the first one is, how much sample does the Fluidity 1M need?
Yeah, so it's for a single data point, it's 4 microliters of sample. And then when we're looking at these other assays, for the labeled probe molecule, we're really looking at a few nanograms for a complete assay. And for the test samples, this would be, for example, a serum or a membrane protein library, it's on the order of 50 to 60 microliters.
Great, thank you. And the second question we have here is, what sample preparation is required?
So essentially nothing. So obviously there is one. So the probe molecule needs to be fluorescently labeled. This is very straightforward very straightforward kind of thing to do these days. And we have very easy to follow protocols for that if it's something you're not familiar with already. And then for the other sample, you don't really need to do anything. So for the serum, it's used directly as it is. For the membrane proteins, we have a protocol for preparing those. But that's one of the strengths of the technology is there's not a lot of upfront preparation of samples.
And then we've got two more here. So the first one is, has this technology been used in any peer-reviewed work?
Yes, it has indeed. So we have something on the order of 70 publications. So the vast majority of those can be found directly on our website. So if you go to our website and click through resources and then publications, you can find those there. And if you have any trouble getting access to any of them, please reach out to us and we'll see what we can do.
Great. Sorry, we've got a few more questions coming in, so I'll try and get them in. So, as a user, what do I have to do to actually carry out an analysis and get experiment recommendations for fluidity insight?
Yeah, so that is really just a matter of running those initial experiments. So we have a kind of, as I say, a standard set of conditions. We provide templates for the run for that, as well as guides, which are all again available on our website. So if you're interested in seeing what the protocol really looks like, our protocols are available on our website. So you run those initial data points. It takes 25 minutes to run the plate, and then the data is just transferred to Fluidity Insight, where the analysis takes on the order of 5 minutes, and then you get on a single page, you get the kind of existing fitting results, which may be even sufficient on their own. And then further down the page, it'll also show you the recommendations for where you need to go afterwards. So it's extremely easy. It's really just point and click on that website there.
Super. And then the last one that we've got that just came in was, what are the lower limits for ligand size?
For ligand size, okay, so the instrument can measure particles between 1 and 20 nanometers in hydrodynamic radius. So what that translates to is 1 nanometer is around 1 to 1.5 kilodaltons. So, sort of small peptides of around 10 amino acids. And at the other end, as we're talking about kind of ranges, there's sort of 15 megadaltons, which is, in terms of particles, it's about an E. coli ribosome, or these sorts of other intracellular macromolecular machinery would be about that end. So, we can cover pretty much most of the proteinaceous type species you might find inside a cell.
Brilliant, thank you. That's all the questions that we've got. So thank you, Sean, for presenting. If anyone does have any further questions, please do email in to the email address that's on the screen, and a recording of the webinar will be sent out to you in the coming days. Thank you very much for attending, everyone.
Thank you.